And what I want to do is to give you an introduction. You may or may not know everything about microbiome. I have to assume that you don't know much, but so hopefully it doesn't look down on you if you know a lot. But I just wanted to be on the same page in terms of microbiome. And so across civilization, in medicine, some aspect of medicine was dictated by shape, size, and texture of feces, believe it or not. And, you know, through centuries, medicine evolved and we get much better. And now what we're using in 1997, we're using the Bristol stool chart, which is a sophisticated way to say that depending on the appearance of your on your feces, you may have some metrics of health that's showing up. And that's, you know, not exactly a very strong diagnostic of any health issues, but I'm just saying over, you know, feces have been used throughout a long time to look at and assess some sort of general state of health. Now, it's not so much the appearance that is important, it's what's inside. What's inside these feces? Well, a lot of microorganisms, as you know, trillions of microorganisms. Actually, if you count the number of cells, human cells versus microbial cells, you get something like almost equal to in numbers. So there's a lot of microorganisms within yourself and on yourself. If you just go by number of genes, okay, so that's a representation of a human. Here's us, 23,000 genes. This is them. A vast majority of us, genes-wise, derive from microorganisms. And if you think about disease, most of folks out there, researchers, they are focusing on human genes. Right? So sequence the genome, do you have susceptibility to XYZ disease? Can we target the genes to have a better health response? So all the space, medical space in terms of treatment and prediction is here. And we're not paying attention that much to this vast array of genes. So why are we doing that? It's a missed opportunities. And the microbiome field emerged a little bit like that. Emerged from the sequencing, the human genome sequencing that created new sequencing platform. And the next step of sequencing was to sequence the entire kingdom of microorganisms inside you. So now we have that knowledge. What do we do with this knowledge is the question. What you think, you know, I was introducing microbial genes and your own genes, us and them, but it's not us and them. We have to view this holistically because we are integrated, all of us, with these microorganisms. Where are they? Are they all the same everywhere? They are not. The microbiome is composed of archaea, bacteria, fungi, parasites, and viruses. And the ecosystem is different whether they are from your skin, urinogenital tract, oral, and so on. So they have their own little ecosystem that are differ with each other. And obviously, the gut. And the gut is probably the headquarters of all these microorganisms. You have something like in a hostile environment like the stomach, which has a lot of acid, not many organisms can survive in this environment. So that's why you have like maybe 100 species in there, not that many. If you go down the tubes, well, there's a lot of resources, all your food broken down in pieces, fair game for these microorganisms. They're going to feed on that. They use this as a source of energy. So you could, that's why they bloom. They have so many, 10 to the 11 or so. So it's a gradient of microorganisms as food get more accessible to them and you have a better ecosystem. What do they do? Why are we getting these microorganisms inside us? Are they just, you know, feeding on us and just without any benefit to us? Actually, they have great benefit. So if you think about metabolic, nutritional, energy function, they provide unique ability for us to thrive, health in health. So they synthesize different vitamins that we can't on our own. Detoxification of compounds that get inside you, very important features. Metabolite production, a lot of small molecule from feeding are shine through your body, and we use it to thrive. So it's a very important function. In terms of immune response, the innate or adaptive immune response is shaped by these microorganisms. They teach us how to respond so that we don't overly aggressively attack different organisms when it comes into contact. So dampen inflammatory response, induce regulatory cells. And also, since they occupy the territory in your gut, they settle there and they prevent enteropathogenic bacteria that you acquired through food or food poisoning or drinking contaminated milk or water. Well, they have a place and they fight for that place. So when this organism come, it's very difficult for them to come and settle because the food is rare and these endogenous bugs that you have prevent them from doing that. So it's very important barrier to infection. Now, I was touching upon the biotransformation or metabolite. So, obviously we come into contact with dietary compounds or nutrition, right? Every food that we eat could be breaking down in pieces, and some of these microorganisms even break them down in smaller pieces. So chemicals and tolerant are also in contact with us, and these microorganisms knows how to detox these guys. We think detox only the liver, but we'll come back to that. Or any meds, actually. Some of the medication we take could be metabolized to benefit, enhance the medication, or deplete the medication. So you will not be responding to the medication. So it's very important to know who is doing what. All these microorganisms are responsible, have a good or an important impact on disease risk, bioavailability of different compounds, toxicity of different molecules, and efficacy of drugs. So think of it like an organ. So when we think about bacteria, we have a different picture of that, right? Some nasty bugs that get you in disease territories. But the collection of microbiome is like an organ. It functions like an organ. And like an organ, often it could be injured. We could injure our organ, we know that. The microbiome is the same, because the microbiome could be impacted by environmental factors, diet, obviously, inflammatory condition. When I talk about inflammation, I'm not saying like rheumatoid arthritis or chronic inflammation like inflammatory bowel disease. Some of us have subchronic level of inflammation. And that type of inflammation is not always detectable, but could impact these microorganisms. The lifestyle, all this make the organ, the microbiome organ, moving in function in good ways, right, to maintain health, or in bad ways to foster disease. So you need to keep balance of your microbiome. How do we do that? That's a learning curve, and that's what we're working on microbiome, to understand how could we reset the biome. Now, this shows my skill. I try to use a software to draw a tree. So it is a tree. Okay? The root are intestinal biome, and branches at the end of all different diseases. All of these diseases were found on the microbiome of case and control. So folks that are healthy versus folks that have any number of these diseases, their microbiome is different, which indicate you may be using the microbiome as a diagnostic tool or a surveillance to see if you could reset the biome to prevent these diseases. And a lot, so this is mostly, correlation studies. Some of them are causative, so you have a causation that the change of biome causes a disease. This is a very difficult task to do in science, to prove a causation. A correlation is right, it's easy. We have a cohort of people, we look at their microbiome, they are healthy versus folks that are not healthy. If we see difference, we say, hey, look, the microbiome is different. But that's not enough of a powerful observation to action on that. How do we get these microbes? Well, actually, vertical transmission. Mother transfer microbiome to their babies, either through a birth canal or C-section. So you will have a microbiome of the skin of the mother. When you're born, as you pass through your germ-free, as you pass through the birth canal or get outside, you acquire microorganisms from the environment. And the only microorganism that you get at birth very early on is this dot here that is green. And if you go here, you have the key. It's a bacteria called Enterobacteriaceae. It's a family of bacteria. Actually, Escherichia coli, which I think you know, is from that family, Enterobacteriaceae. Now, as you evolve through life, the size of the circle indicate the abundance. So this big circle here is Bifidobacterium. You get it from milk. So that bugs will colonize folks at 1 month, and most of it is Bifidobacterium. At 6 months, you start solid food. You start moving around. You start putting things in your mouth. You start to acquire things. Now you develop a very different microbiome than when you were at birth. Now see, this circle is tiny, so you have very few Enterobacteriaceae, and you acquire a different set of microbes. And as you start moving along, by age 3, you're set. Your microbiome will be set from 3 going forward to 75, 80. As you get older, you decrease the microbiome. You will have less diversity in the ecosystem. And that's a study. Those are very important study in terms of neurocognitive and neural disorder. Try to understand to maintain a microbiome when you age. That's a different topic though. But the key here is that microbiomes acquire over time, and you build a mature microbiome that is set, unless you're exposed to repetitive antibiotic treatment, you will be set with your microbiome and should be relatively healthy. The higher level is called phylum in the kingdom. The lowest level is species. So how do we study microbiome? How do we know the name of these bugs? Well, this is sort of a workflow how we do that in the lab. So you have an ecosystem. So if you say, Christian, I'd like you to tell me what's my microbiome, I will say, which organ do you want to look at? Most people will look either at saliva or feces or skin swab, whatever. So we call that an ecosystem sampling. So you have a bunch of bugs. How are you going to get to the information you need? Nucleic acid. Any life has nucleic acid. So now you could isolate from this ecosystem the DNA, right? Or the RNA of this ecosystem. And you could perform different analysis. One key is called 16S ribosomal DNA from bacteria. So what is that? Ribosome is a gene that encodes for some important molecules that are involved in translation of protein. The gene is composed of 8 or 9 variable regions in the DNA. And some are constant. So if you for example, we study mostly the region V1, V2, and V3. So if you do technology like PCR amplification of DNA, we're going to have the entire section of this gene sequenced. And by using informatic tools, so the sequencing is high throughput. This is a technology to sequence. Then we use computer. And then we generate something like that. You will see this in my talk. What is that? This is a spatial representation of microbiome. Every single dot is the microbiome of someone or something. So the entire microbiome is represented here. And the distance between the points shows if they are similar or dissimilar. So here there were two different ecosystems. It's like the right side or the left side of the room, for example. Here, the blue of clustering differently than the red. And so you will call that two different microbiome, these system here. Now, I did that in my lab. People were curious to see the relationship of their microbiomes. It's like if I was to microbiome you guys, I could do that. So what we have done here is to collect feces from all the lab members, and we sequence their microbiome. And again, every single dot is a human being, okay, in relation with other human. The color code means diversity. How diverse is their microbiome, okay? So the brighter the color, the more diverse they are. The more dark or blue, the less diverse they have. So right now you see that number 21 is not very diverse compared to these guys here, okay? Some are babies from or family from these folks, so they have less microbiome. Like I told you, when you're born or young, you have less diversity than older folks, for example. Now, what does it mean, diversity? In an ecosystem, it's thought that if you have a diverse ecosystem, you're healthier. Think of it like here, this is a Christmas tree ecosystem, right? There's only one type of tree. So it's not diverse, not a diverse. Is it unhealthy? Probably not. I mean, if you think about the function of a tree is to fix carbon from the atmosphere, this is very efficient to achieve that task. As opposed to these guys that are bright color, it's like the jungle, many different trees, very diverse and rich. Are they in better health? That's not clear. So every time you do microbiome, you have to compare it to the health status of a person. You know, just looking at diversity doesn't tell you it's great. You have to know different metrics of that person or that organism or anything that you study. Now, how do we study function of microbes? How do we come up with function? If we sequence, it's just a list of genes of microorganisms, like a textbook, but you don't know if it's important. When I was recruited to the University of Florida in 2013, I needed a very specific infrastructures in order to move here. And that structure is called germ-free. So what you have here is isolators. This is a stack isolator, one here, then another one, and so on. Those are filtered system that prevent access of microorganisms. So if you have a mouse that you derive in there, we breed mice, experimental mice, lab mice. And we have them deriving germ-free, and they live in these bubbles. They will never meet any bacteria whatsoever. So they are depleted of microorganisms. And what do I have in these? I have different mice that are, like this one is a knockout of an immunosuppressive molecule. So that mouse is susceptible to colitis. That mouse here, it's an EPC-min mouse. 80% of folks with colorectal cancer has a mutation in that gene, APC. So this is a colorectal cancer model. And here, the cross with AL10 and APC is a model of colitis-associated cancer, colon cancer. If I breed all these mice in the germ-free, they have no disease. They don't develop cancer. They don't develop inflammation. So now I could ask the question, what is the role of microbes if I put that in the mouse that never got microbes? Will they get disease? And you could do that because we have smaller here experimental isolator where we transfer the mouse that we have in a breeding. We transfer them in an isolator for experiment. And then we could put any bugs we want and answer the question, what happened to that mouse when I do that? So we call that humanizing a mouse. I could take your biome, put it in that mouse and see if your biome is healthy to that mouse or be conducive of a disease, for example. Right? And then I could sequence and understand who is in there that caused the disease. I told you that one of the main research aspects in my lab is colorectal cancer and inflammatory bowel disease. So I studied these two diseases, among other things. And one question we were asking ourselves is that we knew that the microbiome of colorectal cancer patient is different than healthy. I remember I told you correlation. Well, disease, cancer, different biome than a healthy person. What do you do with this information? Does it impact the disease? Now I could do, and I can't answer that question because I will collect from tumors of a biopsy patient. I will extract bugs, we did, from a cancer patient or a healthy person, and then use my technology that I told you. We extract the bugs and we're gonna introduce them into a mouse that is germ-free, never saw any bugs, keep them in this bio bubble so they will always be isolated. They only keep the human biome. And then look for cancer development. This is that project was led by Sarah Tomkovich, a very talented PhD student who now works for a pharma company in microbiome. All right, now I put some music to add to the drama. This is colonoscopy, the same way you do colonoscopy in human. This is a mouse. This is the mouse that is healthy microbiome from a patient, healthy subject. Here is my cancer microbiome that I put in the mouse. Look at the tumors here. So we were able to transfer a cancer phenotype just by taking the bacteria from a cancer patient into a mouse that don't develop cancer if they don't have these bugs. The mouse that has the healthy biome did not develop cancer. So it's the type of bugs you have that could cause this disease. And because we could sequence these bugs after, when we isolated, we took the tissue from these mouse and then sequence them for gene expression, which genes is induced in these bugs. And then we saw a lot of genes expressed by microbes that degrade your mucus. Your gut is protected by a layer of slime called mucus, and it's protecting you from bugs coming inside. You don't want them inside you. Well, if you have bugs that degrade the mucus, now you're potentially be exposed to bugs. A lot of motility, so they could breach your epithelium. This is your epithelium from the gut. They could adhere to the epithelium. They resist stress, they secrete toxin, and they are very good in acquiring iron, which is essential for their life. So that all this is indicative of group of bacteria that are aggressive and associate with disease. How do we get to the representation of microbes that could cause cancer? Okay, so this is a cartoon that I use in the lab, try to explain that. So this is you in terms of cells. So you have epithelial cells from different layers, you have immune cells, right? And in your gut, they are producing some compound that are influencing the bacteria. And at the same time, bugs produce compound that impact on you. So it's a conversation. When we're born, we acquire one bug and then build up the microbiome, and we build up health through this interaction. This is healthy. You want that interaction. Now, there's stressor. We never, there's always something in the environment that could push this beautiful relationship, right? Which kind of modifier? I told you about inflammation, diet, medication, and genetics, your own genetics to a certain point. So those are factors that could disrupt this interaction. What happened? Well, your microbiome changed. It's not the same shiny, beautiful one that were helping you. This one changed. It changed in the level of planktonic, so the free in your tube that moves around, or also the one associated with your bowel. We call that an organization microbiome. And that change in cancer, what we push forward as a concept is that this change creates a microbial carcinogenic network. complicated, but at the end of the day, it's these bugs secrete protein molecules that put you in danger of cancer. Which kind of molecule? Well, I will talk about this, but different toxins that are found in human that put you in danger of breaking your DNA into a tumorigenic response. And then you have chances to develop cancer. Let me, and this is different papers that have shown that. These molecules don't work in silo, they integrate and interact with each other. For example, this small molecule here, colibactin, works with this guy, B. fragilis toxin, and that was found in human. These two bacteria are found in human in high prevalence. If you take these bugs, put it in the mouse, cause cancer. So these interactions are very important. I like to focus on this one, the colibactin, because that's one of the molecule that we've been studying for quite some time. It was discovered in 2006, quite some time ago. And it's an E. coli. It's produced by some Escherichia coli. Most of the Escherichia coli are friendly, except enteropathogenic E. coli that gives you, the Turista or the hamburger disease. Those are not good E. coli. That E. coli is not disease producing, but it has an island, a gene island called PKS. And this causes DNA or cell cycle to stop. The cells is not proliferating, it's stopped. And when you stop proliferating, it means your DNA is damaged. Something is wrong, and the cells stop proliferating and try to fix the DNA. Because if you don't fix the DNA, you may have the disease, such as cancer. That was a paper that was published in Science, a big journal. We, in 2012, found, almost by accident, that an E. coli that we call NC101, this E. coli is found in many lab mouse. Many lab mouse have that. No one knew what it was doing. But what we were able to see is that this North Carolina 101 PKS island cause disease, because if we delete the gene from the bug, it doesn't induce as many tumors in this graph as you can see. And if there were tumors, they were smaller in size. So that's an important observation. It means that in this model, inflammation was promoting the E. coli to induce cancer through that genetic island. Later on, folks in Harvard and Yale solve the molecule. So the PKS island is producing this compound that attack DNA. So now we know that this molecule, this bacteria produce a molecule that attack your DNA. And that was a great finding. Another important one is that this molecule that attacked DNA, they were able to find mutation, specific mutation that it was doing on your DNA. And they were tracing down this mutation in humans that have colorectal cancer. So the molecule is associated with DNA damage and mutation in your DNA, suggesting that it's probably an important molecule that increases susceptibility to cancer. Now, you know that cartoon, right? I told you microbial carcinogenic network. Who is influencing the network? How do we get there? I told you the default is healthy, but if there's modifier, if you're exposed to modifier, it transforms itself into a nasty network. How do we do that? Well, I said inflammation and diet. I'll highlight this paper. This is a human cohort of more than 150,000 folks. And what they found is the Western style diet increased. Well, the folks that take a Western type diet and have tumors are enriched for the PKS E. coli that I just mentioned to you. So diet influence the number of E. coli that produce that compound and associated with high risk of colorectal cancer. The other thing I said is inflammation. Inflammation is a big environmental factor, changing the microbiome. Diet, we could control, right? We know that a healthy diet, greens and less of this and less of that, puts you at a better health status than if you don't pay attention. We know that, but it's difficult to change diet. We know that. Inflammation could be actionable. So like the question we had in the lab, if we block inflammation, will we prevent this change of microbes to promote cancer, right? So that, think about aspirin, for example, that has been chemopreventive, associated with some comorbidity though, but it's the same principle. But here, we try to change the microbiome. So I will show you a little story. The lead on that was Dr. Ye Yang. What he did is a very simple model. I told you about the PC mouse, that is a model of colorectal cancer. So we treat them, these mice, with antibiotics just to clean the gut. So we could come with this bacteria, NC101, that has the PKS-ilin. Okay, so we introduce the PKS-ilin. This is a molecule that induces inflammation. And after that, we treat the mice with anti-TNF, which is the same as Remicade when infliximab is in the first line of treatment for folks with rheumatoid arthritis, IBD, psoriasis, and so on. So this is clinically relevant molecule. Now, some mice did not receive treatment and some did. What happened? Okay, the mice that are anti-inflammatory treated have less number of tumors, right? And then this is a picture to show which kind. When you open the mouse, you open the colon, you see the tumors and less tumors in treated. So the anti-inflammation decreased tumors. Great. What happened to the microbiome? Remember the dots that I told you about? So this is the microbiome of all the different mice that were treated or untreated. At day 0 of the experiment, they were not treated. We just started. So there's no difference with the red and the blue distribution. They are all the same because it's the same group of mice. At day 14, this is the 3rd injection of the anti-inflammation. This is the same group of mice followed over time. So you collect the feces from these mice. At day 13, 14, look at the red and the blue. The microbiomes start to separate between who is treated and who is not treated. At day 35, that's the 6th injection of anti-TNF treatment. Look at the microbiome, very different in the feces. And when we collected the mice at the end, this is the tissue microbiome. And again, the difference. So the treatment changed the microbiome. That's in a nutshell. Great. You don't know the relationship of that though. A very simple experiment we do in microbiome is co-housing. So what we do, we will put the mice that are treated and non-treated in the same cage. Mice are coprophagic. They eat each other's feces. We don't tell them to do it. They just do it by nature. So it's not a forced experiment. All right? Just want to make clear. This is, you can't prevent that. And so they share the microbiome passively. Okay, so we did the same experiment. Now they'll eat their microbiome or feces. So the same thing we did before I explained that to you. And now look what happened to the tumor. There's no effect of the drug. The mice received the drug 6 times the same as before the other experiment, but now they share the microbiome. Number of tumors, no difference. Look at the microbiome now. Remember I've shown you that there's progressive change, blue and red. Well, now there's no change. Microbiome did not change because they trade their microbiome. So the drug is there. The drugs target genes inside the mouse, but the microbiome doesn't change. If the microbiome doesn't change, you don't have an effect on tumors. Well, let's go a little bit further. Let's go further and said, okay, let's take the feces of a TNF treated mouse. Take just the feces and the control and put it in a fresh cohort of mice that don't receive the treatment. The only thing that these mice will get is the feces of a treated mouse. Okay, and then do the same experiment, same experiment. And what do we see here? We see that the number of tumors, the only thing that changed here is we introduced biome treated by anti-TNF, put it in the core. And now these bacteria are not able to induce tumors. So you change the microbiome capacity. Remember, the question was, is inflammation important? And I block inflammation. Will I change the microbiome? The answer is yes. Will I change the capacity to induce tumors? The answer is yes. The treatment is not only on the cell of the animal, but comes from also the microbiome. That opened possibilities of treatment management through microbiome. Which kind of manipulation could we do? Well, I told you dietary manipulation is certainly something important, and it's been studied at this point. Prebiotics, probiotics, also a way to manipulate the biome. Not clear though if you could get therapeutic effect. Bacterial therapy, what is that? This is poop peel, right? So this is real. I mean, it's been done in clinic and different treatment now. There's companies, Open Biome, that pays to give shit to a company. Less than 1% of people qualify because they've screened so many things that you have to have the perfect golden feces to be in the bank. And so they don't get Joe on the corner of a street to do that. Okay, it's a very well vetted. So you could, and the peel now are color, so you don't see through. That's the first generation. It's not very appealing if you look at that. Now you could target genes, microbial genes. Then there's drugs right now. You could do bacteriophage. Those are viruses that kill specific bugs. So you could delete specific bacteria in your ecosystem that cause problems, and then eliminate the threat. You could study the microbiome for drug interaction. Is the microbiome good to have my drug interaction? Or you could generate biomarker that predict disease and so on. I'd like to entertain you on something here, which is bacterial therapy. Okay, where is that coming from? There was a paper a while ago published on the recurrent C. difficile infection that is a very, very high concern because of antibiotic resistance. We have certain line of antibiotics, and if you don't respond to the top shelf one, you're doomed. There's no new antibiotics. Companies, pharma don't research antibiotics anymore, right? So you'll be in bad shape if you find a bacteria that infects you that is resistant to antibiotics. This is the case of these folks. All right, how do we save that? So this is the classic treatment, vancomycin or bowel lavage. So you have 30% of efficacy. If you do FMT, fecal transplantation, from a healthy donor, you almost have 90%. This has been repeated throughout the world. This is not like, Rochester's Mayo Clinic that could do that everywhere. And companies are starting, obviously, going after that observation and say, hey, they may be the microbiome, maybe their next source of treatment therapeutics. And this is a head-to-head. Well, I'll skip that. It's too much details. I don't think I need that. All right, so FMT, so powerful for C. difficile infection. And the idea is to replace your bad microbiome, bring a new set of microbes, shiny new microbes into a person to achieve health, okay? For different disease and C. difficile, it has not been shown to be therapeutics. And because we don't know the delivery mode, oral, gastro, rectal, the frequency, which kind of donor. So it's been very difficult. So if people move to second generation, which is a synthetic cocktail from microbes that you think drive the effect, and just use the synthetic cocktail so you won't have a poop pill anymore. Nurses will not be yucky about handling feces. They will just have a clear tube with bugs. Like that, you could drink it. Oh my God. What I've done. So so it's very important research. And the third will be a small molecule produced by these guys. Maybe I could have a drug, right? So this is the template. And all these diseases have been used studies for microbiome, fecal transplant, or cocktail, all of these. Now, go back here, drug interaction. I'm going to segue. The first part was like, oh man, some bacteria will cause cancer, bad, but I want to finish on a positive note for you, okay? A ray of hope that the microbiome could be very helpful for disease. All right, I'm going to focus on something called immune checkpoint inhibitors. Complicated, but the immune system is very powerful, so powerful that autoimmune disease could lead to destruction of organs. So we know that, right? Joint, celiac disease, a different disease that basically, or diabetes, right? Destroy your function of the organ. So they have to be controlled. And a way to control them is by a binding of receptor. Once a molecule binds to that receptor, it signals to the cell to stop its activation and even die. So you remove activated immune cells. Well, the problem is cancer learn to use these brakes. So they use the brake like this. It will engage this molecule is called PD-1 ligand. Cancer cells express that and trick the immune system to say, hey, I'm not there. Don't touch me. I'm a friend. So the immune system doesn't fight it. Now, we could disrupt this interaction. So we use antibody that will bind this. So now the cell doesn't receive this dormant signal. And now the T cell said, wow, attack. And that's how you almost, improve life using these immune checkpoint inhibitor, which earned Nobel Prize to two scientists, one at MD Anderson, James Allison. Okay, now it's beautiful, but very few people respond to the immune checkpoint. If you're a responder, great. I mean, you could have survival for 15 years. Well, you were almost doomed before the immune checkpoint inhibitor therapy. So it's very important, but there's still a lot of non-responder out there. Turns out that I wrote this review capturing three very important papers. What they've done, the same thing as explained to you, case and control. They look at patients that were non-responder of the drug PD-1 and responder patient. Look at their feces and found that the microbiome is different. Nice. Function, well, they put that biome into a mouse that carry the same type of tumors and treat with the same drugs as the human. And what they found is if the mouse carry the biome of a human responder, it will be a better responder to the PD-1 and kill tumors. But if you have the non-responder feces, you're not able. So now it's important because there's something in the biome that could transfer the drug magic. And those are three different groups. These were done in Europe for melanoma. There were no data on non-small cell lung cancer in North America. So we wanted to do that, and we partnered with Moffitt to study that. And what we've done is the same. We have a patient cohort that we collected feces before treatment and after treatment. And once we got the information about who responded to the drug, do they have prolonged free survival, CT scan, less tumors, we have all these data. So now we could link the microbiome with the patient response. The contact person, Moffitt, was Christine Pierce. She has since left for pharma research. And the folks here at my lab, Rachel Newsom and a faculty, Rad Garabay, expert in microbial genomics. Okay, so. Okay. What we did, we took the responder feces. I will we found difference in microbiome from patient responding or non-responding to PD-1. But what I want to show is this. We took the feces from responder patient and non-responder, introduced them to a mouse, the same type of experiment before. These mice are germ-free, so they never had any microbes before. So they are humanized, okay, in terms of microbes. And then implanted a Lewis lung carcinoma cell line. So it's coming from the tumors. And let the tumor grow. And after that, treat with the PD-1 drug and see if we have less tumors in these mice. This is a growth curve. So the non-responder in red, see the tumors are bigger than the mouse that received the feces from a responder. If you don't treat with PD-1, just let the tumor grow, there's no difference. So it's really when the drugs is there that the bugs do their magic, okay? And then we sequence these guys. This is a little bit complicated, but what we did is, this is the feces of human, of 4 different individual responder, and we pull this to give to the mouse. So this is the pool of the human. The same with the non-responder and the pool responder, and we put that in the mouse. Here's the microbiome of the mouse, non-responder and responder, very different biome, very different biome. So now we think there's something in there very important. And when we sequence them, it's a little bit complicated, but individual dots here represent different species from this genus. Above the red line means enrich and responder. So these responder patients have all these type of bugs. I tell you something, you can't, there's beautiful genomic, but I don't know how to action on that. I need the bugs. I need the bugs inside these patients. I need to have it. That's very difficult. We partner with a company, and we're very lucky to have them because they don't sell that technology. This is a what we call a prospector, nice name. So you basically mine feces. We put that machine inside a bubble, which means that this is anaerobic. There's no oxygen. 95% of the bugs living in your gut don't tolerate oxygen. They will die. So you need to keep this free of oxygen, okay? And the technology has 6,000 nanowell, individual nanowell that we could spray feces in the array so that we have individual bugs by calculation in each well. So bacteria don't compete with each other. And we have a marker, a fluorescent marker that tells you if there is something growing in that well. And then with a computer detection and pickup, we could array bugs, individual bugs. So we isolated 1,800 bacteria from feces of responding patients. And then to identify them, we use a technology, MALDI-TOF. Those are, chromatography type. And then if we don't have it identified, we sequence the gene. So bottom line, we identify 183 bacteria, unique bacteria that we could survey for function. Which kind of function are we looking at? I'm almost done here. We look, I told you the T cells are important to kill tumors. So the screen, what we were looking at is the capacity of effector T cells to produce this molecule that is anti-tumors, interferon gamma. So all the single isolate we obtained from the machine, we grew them back in anaerobic. We filtered the soup of the growing so that we have only a supernatant. And then we test screen against this molecule. And this is a graph that shows you that. This is how much interferon gamma. So this is our positive control. This is a bunch of bacteria that do not have any effect. But here, this is 6 bacteria that induce interferon gamma. So we said, hey, I think we got it. I think we got the bugs that will be anti-tumors. So what we did, we created a cocktail. We have this cocktail of 6 bugs that induce interferon gamma, anti-tumor from T cells, and we put it in the mouse, right? to see if they will kill. Now we move away from feces to 6 bacteria that could grow. We could grow them, and did the same experiment. And this is what you have here. Now, this is the responder feces with anti-PD-1 treatment, so it's here. And the non-responder feces, the same experiment I've done before. And now look, this is my consortium, $6. It's just as good as the total feces to kill tumors. So now I have a magic bottle concoction that I think I could use to help patients fight tumors with this therapeutics. So I'll sum up this for you. I hope it was not too much of information. The first potential important aspect of this lecture is that the carcinogenic potential of some E. coli could be attenuated if you block inflammation. Okay, so I open this is treatment of microbiome to prevent disease. That's what it means. Here, transfer of human microbiome from responding patient into clinical model enhanced the drug effect, anti-tumor effect, and an engineered human bacterial consortium coming from the feces of this patient could be used to enhance drug treatment. So we move away from complex feces, a lot of stuff in there, to very defined microorganism. What's the point of that? This last final slide. So this is, we know precision medicine. Precision medicine is about finding who in a cohort respond to a drug, right? We know that a medication don't have the same effect across people. Some are good responders, some will have no response to the drug, and others will have, they will be more sick. So we need to match the pill with the right people. What I think I'm trying to convince you here is that your microbiome is also very important in the decision-making process of precision medicine, because it could impact the way you respond to a compound, and you could customize the biome to make sure it fits you in terms of health and disease. So that's the way of the future, if you ask me. I'll stop here because I think your time is valuable. So there's a lot of people involved. I think I highlight pretty much everyone, yes. And fundings that were instrumental to support that research, because it costs a lot of money to do that. And I welcome your time. I'm very happy to interact with you. And if you have questions, then we could exchange a little bit. Thank you. The summary screen, yes, sure. I think we're recording that. The mic.
Yeah, it's just, I don't know. I'm sorry if that.
Yeah, I think it's broken. That's all right.
How do you do with Alberto?
Alberto? Ah, you know Alberto, yeah, he's genomic.
His father's my brother.
No way.
Yeah, you know.
He's a great guy. He's a great guy. No, I have him on the paper that we submitted. No, we have a great interaction. It's a very knowledgeable person in genomic, yes.
You want to stop the screen share, that way
Stop.
people can still see you too.
Oh, okay, sure.
You mentioned 4 potential modifiers of that. I was
Microbiome.
confused by the third one, medication.
Yeah.
Is that medication in a positive way, the cocktails, or a negative that we are taking prescriptions?
Yeah, medication, if you take different medication, they have an impact on the biome. So, because they could serve as source of carbon and sometimes sugar. So the medication will have nutritional impact on the biome. So they will change their constitution, their composition, I mean. And the medication could also be transformed from these bacteria. So if you take aspirin or mesalamine, they will change the biome. If you take NSAID, anti-inflammatory, will change the biome. Metformin, change the biome. So there's a long list of medication that change the biome. I mean, these changed, what's the consequences of these changes? If they change just for the sake of changing, it's okay. But if they change in a way that you feel that you're worse shape than before, then, that's a red flag. And, but these research, the clinician doesn't have information about that. He cannot tell you that, oh, your microbiome changed that way. I got a test back and here's your microbiome. You need to change X, Y, Z. We don't have the capacity to do that at this point. It will come, but we don't have that information. But from studies, a cohort of people, we see these differences. Yeah.
Get a long list of fine print in every medication, but I've never seen anything say this might change your life.
You're correct, and it will be put on by FDA if they see that there's an adverse effect, and if they don't, it's not reported linking to it, they won't do it.
You said early on that by the age of 3, your microbiome is set. But it sounds like subject to change from these kinds of modifiers, or I got that right?
Yeah, correct.
And then when you take antibiotics and you kill off some of that to bounce back.
No, they do. But if it's a chronic exposure, so, anecdotal, you take antibiotics regimen for 7 days and you don't take anything in the next 5 years, it's fine. But it's especially for kids when they have repetition during their formative years of microbiome, these change may be imprinted and set in a bad way. There's a lot of studies about early exposure to antibiotics and consequences on health for kids. For adults, we tend to be proactive, right? So if you take antibiotics, we'll say, oh, I'm going to take probiotics to help my system, and there's really no data for that. The thing about probiotics that worries me the most is this is a supplement. It's not, you don't know what you have. You don't even know what's in the damn thing. No one knows. We study some of them, and we study one called VSL3, very popular for patients with IBD. We gave that to a mouse and it enhanced cancer. So that was not good. But others that have no effect, because we don't even know if they are alive, so no one knows. So I will say prebiotics is probably better, kefir, yogurt, fermented food. Those are a natural way to change your biome. But if you take antibiotics and then introduce billions of bugs that are only 1 or 2 type, you decrease diversity, and it takes longer to rebuild where you were before because you have introduced so many other things there. So my advice would be not to do that.
Not to take any probiotics.
Not for correcting antibiotics exposure. Yeah.
Thank you.
Yes.
This is very fascinating.
You could search that. There is a landmark papers published in a journal called Cell. This is a very top premium journal, and there's 2 papers on human cohorts with and without probiotics after antibiotic treatment. And all the folks that enroll in that and took on probiotics, their microbiome did not regenerate the way it was, or it took longer than someone who did nothing. So, yeah.
How do the prebiotics change the microbiome? If they create the solid forms and systems?
Yeah, those are fermented food. So, like I said, when you introduce that in an ecosystem, the bacteria feeds on a lot of these nutrients. So if you take complex non-digestible fibers, that's a great way to boost bacteria that are very important for secretion of healthy metabolites. So complex fibers that we cannot digest, but they do. They generate tons of molecules from that. And prebiotics, fermented food is the same. They will have specific molecules that the bugs could utilize. And if they are fed, they multiply, right? So they gain traction. There's more of them, and they produce compound. For example, if you have folks that eat vegan versus omnivore, their output of molecules is very different because the type of nutrients you give yourself is different, and the bugs will respond to that by producing unique set of molecules. I'm not saying that it's healthier. You know, typically a vegan may be healthier than omnivore. You know, the debate is still there, but at the end of the day, there is a change in biome functions. So prebiotics will have an impact on these bugs. Yes. Yes.
Did you say in your lecture that as you get older, the diversity of your biome decreases?
Yes.
And what's the impact of that?
Yeah. I'm paying attention because I'm not getting younger myself. So there is a lot of interest in linking early onset of neurodegenerative diseases, you know, Parkinson, Alzheimer, and dementia that may be linked to microbes. The gut-brain axis is extremely vibrant in research, and we don't know exactly which bugs we're losing and which one was important to maintain cognitive health. We don't know that. We know that if you transfer microbiome from you know, Alzheimer patient into a mouse, germ-free, they will exhibit sign of the same disease. But we don't have clinical actionable item to do correct things like that. But the research is going forward, and I tell you, there's a lot of money involved in that space because it's probably the most challenging healthcare issue we'll face, you know, because everyone pay attention, live longer, and now we discover a different set of disease that are affecting our population, our elderly. So we need, you know, if it's preventable through some microbiome action, that's great. On the other side, sentinel, a super sentinel, people who live 100 years and more, they have a very unique microbiome. Yeah, their microbiome is super microbiome actually. So that's something that, again, you know, they may have escaped this depletion of diversity, and they were able to maintain something that they live healthier and longer.
Don't they eat a lot of yogurt?
I have no idea. I probably do.
On Thursday, we have a course on super agers, so I don't know, it's
Yeah,
part of our life in the extreme, so we'll have to see what they say.
yeah, no, absolutely.
Yeah, I understood that, if you eat yogurt or other things, it doesn't get the bugs so good through the stomach because it's such a hostile environment.
Yeah.
So in a sense, it doesn't matter what you.
Right. No, I mean, I get your point. And actually, that's one aspect of probiotics and delivery of cocktail. You need to formulate to resist the harsh acid environment. But remember, some of these bacteria, lactobacilli, is a classic probiotics, bifidobacterium, and they produce lactic acid. They are used to acid. Yeah.
I see.
Yeah, they go through. They just don't colonize, but they go through. But some maybe you may lose, you know, if you get 100 million you inject and 1 million go through, it's fine. You lost 90%, but 10% is enough to repopulate the entire. Most of probiotics, though, they are not colonized, they're just passing through. Yeah, they are not very good at setting shock. You know.
Do you take any probiotics?
No. I don't want to influence anyone here, but no, I don't. One main reason is that you just don't know what you're buying. There's no quality control. I used to do a lot of research on herbs and supplement, and it was the same, you know, because it's not regulated. So you buy things that you're not 100% sure what match what's written on the label. So I'd rather stay away from it. But like I said, I mean, if there's a track record of safety on it, and in your mind it thinks that it's helping you, by all means, you go ahead. Right? I don't think that I will say you put yourself at risk. I don't think so. It's fine. some folks are in bad shape and things that probiotics will fix them. And this is where I will raise a red flag on that because it's probably not the right approach.
Where do you see the speed leading to? It does get to the point where you figure out these things. How do you think we would get treatment?
Yeah, no, I mean, like I said, yeah, you could, the slide I was showing with, the different way of intervening. You could engineer the microbiome to give you the health benefit that you need. So that probably will come for disease treatment. Not sure prevention, but disease treatment, probably. Markers of efficacy for treatment, for example, or prediction of disease could be, I'll take, a sample of your saliva or feces. I will say you have a high risk of developing XYZ, and we will recommend something. That's something we don't know, but it will come to that. I think there is drugs been generated. We have compounds from this cocktail. We identify a compound, unique compound that was not found before. So we're testing that compound. So now we will have a compound that we could potentially give patient to have a better response to the treatment. And I think it could be applied to brain-gut axis, heart disease, diabetes, everything that is associated with biome. You may find a molecule or a group of bacteria that could help maintain health or diminish progression of disease, for example.
And you may have just answered this one, but have there been clinical trials beyond the C. difficile?
Yeah.
Fecal transplant.
Yeah.
Work.
Yeah.
Have there been any kinds of clinical trials where people have tried to sterilize the gut and start
Right.
a fresh microbiome by putting in?
Right. Well, the FMT is that actually. So that's why we need to work better for implementation for the microbiome to implant in the recipient person. But they give antibiotics before, so they clear up the room for the new bugs to come in. And that has been done for different, inflammatory bowel disease, diabetes, it's been done. The outcome is not clear. There's a couple of papers published in high impact journal like Nature Medicine. So if you take these immunotherapy drugs, your T cell, effector T cells, immune cells are very active at killing tumors, but the collateral damage to give you colitis because they are so activated. You don't have a break on them and you develop, IBD. And so that's an adverse effect. But that could be fixed by fecal transplant. They did that. Patient that have adverse effect, immune adverse effect, they will have fecal transplant, and they had decrease of the adverse effect. They were not responding to steroid immune activation, but they were fixed by transplant. So I think you're going to see that more and more. The clinical trial for C. diff is just the flagship of everything. We don't even have a drug yet for C. diff. We don't have it. We don't have the bugs cocktail yet. We don't have the bugs cocktail yet. So,
well, I think the problem is that we don't know what's in the microbiome. We do it in a general sense.
Yeah.
But I mean, it's an incredibly complex,
right,
three-dimensional population.
Yeah.
And so,
absolutely.
Except at the gross level, it's really hard for us to sort of specifically try and replace.
Well, you raise a very important point. I mean, there was a clinical trial, small pilot. What they did, instead of fecal transplant, they filtered the feces to cut any bacteria. Gone. So the only thing they have is maybe metabolites and viruses. And they took that liquid and put it in the C. diff patient. And they achieved the same effect as if for fecal transplant, which now you think it may not be bacteria, it could be bacteriophage that heal. Right. So there's,
I started with fish, so.
right, so, right, I think we speak the same language here, yeah, so, yeah, we don't know, so these companies that build cocktail may not be on the right track. You know, could be something else, that's the problem, that's why we wanted to move away from the fecal transplant, get the bugs, if we have the effect, what is it that they're doing? And then you build a better case, for you, you know.
Any more questions? Anything on Zoom that you see?
Oh, Zoom, I didn't look.
No. Is there something?
No, no, it's quiet on Zoom.
So thank you very much.
Ah, my
Christian.
pleasure, thank you. Thank you.